Hi, my name is Daniel Blackburn, and today we're going to talk about macronutrient deficiency for plants. And this is the practical where we submit our plants to macronutrient deficiency and see what happens to them. All right, so this is part of the course SWAE4401 Water and Nutrient Cycling in Soil Plant Environment. And this is going to be a two-step video where we're going to, in the first part today, we're going to talk about the effect of macronutrient deficiency on plant growth and biomass. And the next lecture, the next practical, which is part of the same experiment, I will show you what the plant analysis, what happens with the nutrient concentration in the plant tissues. So I will explain a little bit what was the experiment that Lilwa did for us. The experiment consisted this year that we have a sandy soil, a sandy loam soil, where we have very low concentration of nitrogen, phosphorus, and potassium, almost in a very extreme deficiency of nitrogen, phosphorus, and potassium. Then we calculated the amount of nutrients needed for appropriate plant nutrition in this situation. You're going to receive all that on the lab handout. And we have a treatment that receives NPK as a treatment that receives phosphorus and potassium, but not nitrogen. This is the -N. You have a treatment that receives nitrogen and potassium, but not phosphorus. And you have a treatment that receives phosphorus and nitrogen, but not potassium. Okay, so the source of potassium here is potassium chloride. The source of phosphorus is triple superphosphate, and the source of nitrogen is urea. Okay, so either you have the supply of all three of them, or one is missing on the system. So we are going to see what happens with the plants when one is missing. So these plants were held in our patio for 2 months, actually about 1.5 months, and this is what you see from the plants. They're all mixed up here. It's hard to see which one is which, but the bigger ones are the ones with NPK, and the smaller ones are mixed, are the ones which are deficient in nutrients. You can see right away that there is a lot less biomass in growth when you have missing one of these macronutrients for the plant. So once we harvest the plants, we can see here that the symptoms of deficiency, the nitrogen was really clear, the yellowing of these beans. This is a Vigna bean, so we don't see a lot of nodules being formed if the association with rhizobium was successful in this legume. You probably see that some of this nitrogen could have been supplied by the symbiotic association between the plants and this bacteria, but it was not the case here. We didn't have nodules forming, and the plants were nitrogen deficient. So this is showing the color, the difference of color of that we have from the NPK plants and the ones deficient. And Lilwa here is measuring the length of these plants, the length, shoot plus root, and then individually only the length of the shoot length and the root length of the plants. And then you will receive this data showing how big was the plants when you have sufficiency or deficiency of these nutrients. An important thing that I want you to calculate when you receive the spreadsheet with this data is that I want you to calculate the root to shoot ratio. And you can calculate that using the biomass and also the length. Yeah. So what you will see is some nutrients will have a huge impact on root development and other nutrients not, and that will be reflected on a different root to shoot ratio. If you have that there is a high shoot to root ratio, it means that there's more shoot than root. Yeah, if you divide one by the other, you will see that one will be much bigger than the other. And this, if it's maintained the proportion between the sufficiency conditions into the deficiency, it does not have an effect on rooting, just in a direct effect on biomass. But in the case where you have this deficiency, you might have that you might find that this shoot to root ratio is changing. Yeah. So here is just showing Lilwa that she's cutting the shoots and the roots, and this will go for drying, and then you will have a moisture content in the dry weight of this. You have fresh weight and you have dry weight of this plant material, and this is all data that you're going to receive, and you're going to have to show that in your report for the different treatment. So this is now Lilwa showing already cut the different treatments. This is the NPK minus N here. And she will show the other treatments also, how much smaller the plants are, minus P and minus K. So you can discuss the general aspect of these plants, and also with the data, make the graphics to the difference in biomass that you have between the plants with NPK and the ones with the specific nutrient deficiencies. Right, this is Lilwa showing again the measuring of these plant materials. I will just jump a little bit further because there's not much to explain here. It's just showing the length of different parts. You will receive the data. And after we do that, we have cut the plant material, put them in crucibles, put them on the oven at 60 degrees for a few days, and then after a few days, we can when there is a constant weight, this weight is not changing anymore, then we can assume that this is the dry weight of the plants. Before we go for the furnace, the furnace in the nutrient analysis, the ash content, we're going to explain on the next video. Yeah, on the next video. Up to this one, you are only going to receive the data from biomass, and then you're going to discuss the biomass data from this experiment results. But I wanted to show you what we did in previous years also using sorghum seeds. So this is what the experiment that was conducted in sand and using Hoagland solution in sand. And you have here on the right the NPK, and you have here the potassium plants completely died on this trial. The minus P is here and the minus N here. So I will show you also what happens. This is the harvest with the students lining up the plants with different nutrient deficiencies. Each group received a different treatment, and now they're gathering all the data together to share and write the reports. So you can see here the general aspect of these treatments. I will even move my camera a little bit so you can see that in the NPK you have very good biomass development of the plants. Minus N, not so much. Minus P, also a little bit less. In potassium, the plants completely died, and we have that the roots were very damaged on the potassium treatment. So this is just a way to show you same type of experiment. What happens if you have a dicotyledon or a monocotyledon plants doing this? This serves for doing a very practical demonstration of what is the effect of nutrient deficiency on these plants. So for this practical for this week, you will receive the spreadsheet with the data from root length, shoot length, fresh weight and dry weight from different plants, and then you will calculate if there is a significant effect. You will perform ANOVA on this data, and you will calculate and show me on your report what is the effect of the deficiency of nitrogen, phosphorus, and potassium on plant growth. In the control here, the positive control is the NPK treatment. So every treatment you should compare with the NPK and see if there is a significant difference between individual treatments and the NPK. Next week, we're going to show the same experiment, but now the plant material analysis, and we're going to analyze the ash for potassium and phosphorus, and we're going to calculate what is the concentration of phosphorus and potassium on the plant material, and then you're going to make your report on that also. All right, thank you very much, and I see you in the next practical next week. Thank you.